Forensics teams currently work out when someone has died using their body temperature, which falls by 1.5 degrees centigrade, 2.7 degrees Fahrenheit, an hour. However, researchers, led by the Center for Genomic Regulation in Barcelona, stress that their findings have not yet been proven to work beyond 24 hours after death. Lead author Dr. Pedro Ferreira, from the University of Porto in Portugal, said, "We found that many genes change expression over relatively short post-mortem intervals, in a largely tissue-specific manner." This information helps us to better understand variation, and also it allows us to identify the transcriptional events triggered by death in an organism. Changes in the genes begin as soon as someone dies, with the most obvious ones seen between 7 and 14 hours later. While there are few immediate changes in areas of the body such as the spleen and the brain's cerebellum, big changes are seen in the muscle and colon, for example. They identified 187 important genes after death, including the HbA1 and HbA2 genes, which transport oxygen from the lungs and change their behavior after someone stops breathing. How can genes pinpoint the time of death? Genes may help pinpoint a crime victim's time of death, scientists claim. Researchers recently looked at 9,000 samples from 36 different types of bodily tissue from donated cadavers. For each sample, the scientists knew how long had passed between death and the preservation of the body. Researchers found that after death, different tissues responded in a wide variety of ways. Some genes, for example, can continue to work for up to 4 days in a last-ditch attempt to save their cells, despite the death of the host. Some genes can continue to work for up to 4 days in a last-ditch attempt to save their cells, despite the death of the host. To find out how gene expression changed once a person had died, the research team compared blood samples ante-mortem, before death, and post-mortem, after death. Although it isn't ready to be used in the field yet, the levels of RNA were found to be consistent across several different corpses. Scientists believe they can use these readings to predict time of death to within an hour. However, researchers, led by the Center for Genomic Regulation in Barcelona, stress that their findings have not yet been proven to work beyond 24 hours after death.